Hello, thank you for joining us today to talk about Genetics 101 and how to read your genetic testing lab report. My name is Julianne Savat, and I will be providing you with an overview of some of the common terms you might find on your laboratory report, and then my colleague, Rebecca Smith, will be actually walking through an example of a laboratory report to help you sort of put all these pieces together. So today I want to begin with an overview of genetics, and I know that for some of you joining us, these concepts and terms might be familiar to you. For others, these might be new terms to you, or this might be a bit of a refresher from whenever you met with your physician or genetic counselor. My hope is to review chromosomes, DNA, and genes to discuss types of genetic changes or variants, types of genetic testing that you might have gone through, and then some other common terms that you might wish to know or that you might see on your laboratory report. And then, as I mentioned, Rebecca will walk through how to review your laboratory report using an example to walk us through all of these terms. One topic that we won't be touching on today, but that we get a lot of questions about, is gene therapy. Dr. Wendy Chung's plenary presentation on August 14th at 2 p.m. will provide much more information about gene therapy, so be sure to attend that presentation if you have questions around this topic. But now on to an overview of genetics and some of the terms you might see on your testing reports. All of you listening have gone through the genetic testing process, and you might have spoken to a genetic counselor or geneticist. So as I said, some of this might be familiar, but my hope is to break it down and to help give you a bit more information as you re-review or walk through your testing report. Each one of us has more than 1 trillion cells that make up our bodies. We have different types of cells, including our skin cells, our brain cells, our heart cells, and these really are like the building blocks of our bodies, sort of like how bricks make up a house. In each and every one of those cells, if we crack it open and look inside, is our genetic material, or our DNA. And our DNA is like our body's instruction manual. Each instruction is called a gene, and in fact, we have so many instructions, so many genes, that I often say that our genetic material is like a shelf of instruction manuals. To keep our DNA organized in our cells, it's wrapped up very tightly into these structures called chromosomes. Typically, people have 46 chromosomes that come in 23 pairs. Our chromosomes come in pairs because we get 1 in each pair from our mother and 1 in each pair from our father. The first 22 pairs of chromosomes are the same in males and females, and they're really organized based on size and named based on size, with the first pair being the largest down to the 22nd pair, which is the smallest. The 23rd pair of chromosomes, that last pair, are called our sex chromosomes. Males are typically XY and females are typically XX. As I mentioned, each of these chromosomes is made up of DNA that's wound up very, very tightly, almost like a ball of yarn. Our DNA is what provides the actual instructions for our body, and it's actually instructions for how our body can make proteins. Each instruction is a gene, and each gene codes for a specific protein. We each have approximately 20,000 genes that we inherit from our mother and father, and these help our bodies grow, develop, and work. Each protein has a specific role, or maybe a couple roles within the body. Some determine our eye color, others help our heart grow, and others are important for how our brain develops. Our body spells out the instructions in our genes using the letters A, T, G, and C. And our body actually reads through those letters to use our genes as instruction to make those proteins. When reading the letters within our genes, our body actually reads those letters in sets of 3. We call that the triplet code. A set of 3 letters in our DNA codes for 1 amino acid in a protein, and amino acids are just the building blocks of a protein. So a protein is made up of multiple amino acids, and our body would read 3 letters at a time to make 1 amino acid. So in this example, our body would read ATG to make this first amino acid, the letter CAC to make the second amino acid, and so on. There are 20 different amino acids that make up our proteins, all coded by these 3-letter sets. So now that we've talked about DNA, chromosomes, genes, and proteins, let's talk a little bit more about genetic variants and what they are. Genetic variants are changes in our DNA. While most of our DNA sequence is the same from person to person, so myself and Rebecca, most of our DNA is the same, we all have genetic changes that make us who we are. Some variants just help make us who we are, make us individuals. Others affect how proteins work in the body and might cause developmental or health differences. There are 2 main categories of genetic variants that we want to focus in on and talk a little bit more about today: copy number variants and sequence variants. Starting with copy number variants, these are genetic changes where a person has a large piece of missing or extra DNA. It's often thousands of extra or missing letters within their DNA. If a person is missing genetic material, that's called a deletion. If they have an extra piece of genetic material, that's called a duplication. So if we think about our genetic material, our DNA being a shelf of instruction manuals, it's almost like a copy number variant is almost like a person is missing books or has extra books within their DNA, their genetic material. Copy number variants are typically named for their location or where they are along those chromosomes. So for an example, we might see a report that says 6p24.1 deletion. This helps us know where in a person's genetic material this missing piece of DNA is. The 6 tells us that this missing piece of genetic material is on one of this person's number 6 chromosomes. When we talk about chromosomes, we divide them into 2 parts, the P arm and the Q arm. The P arm is the shorter arm, the Q arm is the longer arm, and they're divided by this little pinch point here, or what we call the centromere. So 6P just tells me we're on the smaller arm on the chromosome 6. And then the 24.1 tells us exactly where we are along the chromosome. You can see that the chromosomes have a banding or striped pattern, and this is actually what they look like under the microscope. So the 24.1 tells us exactly what band or stripe has the deletion. So again, copy number variants help tell us where that missing or extra piece of genetic material is. Deletion tells us that a piece is missing. Duplication would tell us that we have an extra piece. The other type of genetic variants I want to focus in on today are sequence variants. Sequence variants are genetic changes where there's a difference to the letters within a gene. Sometimes this is a single letter or maybe a couple of different letters. And if we think about our genetic material as that shelf of instruction manuals again, a sequence variant is like there being a spelling change in one of the sentences in one of the books. So a small change, just one or a couple of letters that's different. Sequence variants are typically named based on the change to the DNA and the change to the protein. If you see a C period or a C dot on your report, this tells you what the change to the DNA is. That C period is often followed by a number, so this tells us exactly what letter is changed in our DNA. So in this example, we're talking about the CSN-K2A1 gene, and we're talking about letter 593 within that gene. So if we start at 1 and count up to 593, that's where we're talking about. The first letter, A, in this example, is what letter people typically have at position 593. The greater than symbol is our way of saying is changed to, and then that second letter, G, tells us the letter in this individual. So altogether, this tells us that at letter 593 in the CSN-K2A1 gene, most people have an A, but in this person, they have a G. This genetic change can also be described by the change to the protein. So if you see a P period or a P dot on your report, this is describing what happens to the protein or what change there is in the protein. This P-lys-198 arginine tells us that an amino acid 198 in that protein, so if we start at 1 and count up to 198, that's the place where we're looking. There's typically a lysine, and this is the abbreviation for lysine, one of the 20 amino acids. But in this individual, they have an arginine instead. So if you're reviewing your report, if you see that C period or that P period, this tells you whether you're talking about the change to the DNA or the change to the protein, and you can break down those pieces after that C period or P period to know a little bit more about what the genetic change is. There are several types of sequence variants that I want to mention and talk through, since you might see these terms on your laboratory reports. The first is missense. Missense variant means that one amino acid, one of those protein building blocks, is substituted for another based on a single letter change to the DNA. So in that example that we just walked through, the amino acid at position 198 in the protein is typically a lysine, has been changed to an arginine. One amino acid was changed. So this is an example of a missense variant. Another type of sequence variant you might see is a nonsense variant. Like missense variants, nonsense variants are caused by a change in a single letter in the DNA. Here we can see that at letter 265 within the SETBP1 gene, most people have a C, and there's a little error here, forgive me, this person has an A instead. In addition to coding for amino acids, the 3-letter sets in the DNA also help our body know when to stop a protein. So sort of like a stop sign that says this protein's done, stop making it. A nonsense variant happens whenever the DNA change results in an early stop sign or an early stop signal. So the protein is shorter than typical, and this can impact how the protein works in the body. If you're looking at your report, you can tell if it's a nonsense variant in that P period or that description of the protein change. There'll be either a TER, short for termination, a star, or an X to note that there's an early stop signal. You also might see sequence variants that result in a small number of extra or missing letters within the gene. Insertions or duplications add a letter or several letters to the DNA sequence, like this example here, where there is at position 6073, we typically have an A, and this person has a duplication or an extra A inserted there. A deletion removes a letter or several letters from the DNA sequence. Now, sequence variants that have insertions, duplications, deletions are different from those copy number variants that we talked about a few slides ago, because these only involve one or a small number of letters within the DNA, while those copy number variants that we talked about often involve thousands of letters within the DNA. So to help sort of make that distinction there, it's just how many letters are involved. Adding or removing letters in the DNA sequence can result in what we call a frame shift variant to the protein. As we talked about earlier, the body reads that DNA sequence in sets of 3 letters. If we duplicate just one letter, like in this example, it will shift how our body reads the remaining letters and can change the remaining amino acids in the protein and eventually lead to an early stop codon. You can identify frame shift variants on your report because they typically have the letters FS in the description of the protein change. Those insertions, duplications, and deletions of one or a few letters can cause these frame shift variants. So now that we've talked about genetic variants, so copy number variants and sequence variants and some of the different types of sequence variants, what sort of genetic testing might be used to identify these changes? All of you listening in today likely are familiar with one or more of these tests from your own genetic testing journey. And all of these tests are typically done using blood or saliva samples, as you might know. The first test that I want to talk about is karyotype. A karyotype is a test that we used to use quite frequently, but we use a little less often now. And it's a test that actually looks at the chromosomes under the microscope. It can identify large copy number variants, so large missing or extra pieces along those chromosomes, or even a whole missing or a whole extra chromosome, like an extra chromosome 21 that causes Down syndrome. So this is sort of the most zoomed out view of a person's genetic material that we can take. If we zoom in a little bit further, we can do a test called a chromosomal microarray. And this test can detect smaller copy number variants, so smaller missing or extra pieces compared to the karyotype. If we want to actually read through the letters of someone's DNA, we have to zoom in even more and use a technology called sequencing. Gene panels use sequencing to look at variants in a specific list of genes that are tied to certain health conditions or developmental differences like seizures, autism, developmental delay. Another type of test that uses sequencing is called exome sequencing. Exome sequencing uses sequencing to look for variants in all parts of our genes that provide our body with instructions. These parts of genes that provide us with instructions are called our exons, and if we look at all of them, we call that our exome. So exome sequencing looks at all of those pieces of the body that provide us with instructions for our proteins. So whenever you have genetic testing, if you identify a genetic change in a specific gene or a copy number variant that involves maybe one or a few genes, the next step for the genetic testing laboratory is to interpret that variant or to try to determine if it impacts how the gene works in the body and if it could be associated with a health condition, a developmental difference, or a risk for a condition in the future. As I mentioned earlier in my talk, we all have lots of genetic variants. Many of them just make us who we are, and only some will impact our health and development while others don't. So when a lab sees a genetic change, they try to use all of the information that they have available to them to try and determine if that genetic change might be associated with health or developmental differences. This means that they look at things like, how does that genetic change impact the protein? Does it cause a frame shift? Is it a missense variant? They look at where it's located in the protein. Is that a piece that's important for that protein's job in the body? They look to see if that genetic change has been seen in other children with similar symptoms or health concerns. They look to see if it's been seen in the general population. And then they try to put all of this information together to assign an interpretation to that variant or what they think about its relationship to health. Interpretations from genetic testing laboratories primarily fall into three categories: benign, uncertain, and pathogenic. Benign means that the copy number variant or sequence variant is not known to cause any significant health or developmental differences. Pathogenic means that this copy number or sequence variant is believed to cause symptoms or features. If there's not enough information to put a variant into those categories where we say, yes, it's tied to health or development, nope, we don't think it's related, we have to call it uncertain. If we don't know enough to say one way or another, we say that's a variant of uncertain significance or a VUS. This means, again, that there's not enough information for us to say if the variant is tied to health or development and that more information is needed. Our hope is that over time, we'll be able to better understand that uncertain result and to put it into a more concrete category, but sometimes that just takes time. We need to learn more. Other times we can recommend or offer things like parental testing to help inform uncertain results. If parental testing or testing mom and dad wasn't done as part of initial testing, this can help clarify things in some cases. Understanding if the variant was inherited from a parent or if it's new and unique to the child can help us better understand that variant and its link to health or developmental differences. So on to some other common terms you might see on reports. So first, on your reports, you might see the terms autosomal dominant, autosomal recessive, and X-linked. To walk through these terms, I wanted to first return to this slide that you saw a little bit earlier today. As a reminder, we all have 2 copies of the first 22 pairs of chromosomes. We have 2 copies of each because we get one from our mother and one from our father. So for all of the genes that are packaged into these 22 chromosomes, we typically have 2 copies of those genes, one from mom, one from dad. Again, our last set of chromosomes are our sex chromosomes, with females typically having 2 X chromosomes. So they typically have 2 copies of all of the genes that are packaged into those X chromosomes. And males typically have an X and a Y, so they have 1 copy of the genes that are on that X chromosome. Autosomal dominant and autosomal recessive are used to describe conditions caused by changes in genes that are on those first 22 pairs of chromosomes. Conditions that are caused by having a pathogenic variant in 1 copy of the gene are called autosomal dominant. If you need to have a pathogenic variant in both copies of the gene, this is called autosomal recessive. Conditions caused by pathogenic variants in a gene on the X chromosome are called X-linked. Symptoms can differ between males and females because females typically have 2 copies of this gene, while males typically just have 1 copy. They only have that 1 X. Sometimes autosomal recessive, autosomal dominant, and X-linked are called inheritance patterns. But these alone don't tell us if a genetic variant was inherited from a parent or whether the variant was found on 1 or both copies of the gene. They're really just used to help us understand if having 1 pathogenic variant causes a condition, or if you need to have pathogenic variants on both copies of a gene to cause a condition, and whether the gene is located on those first 22 pairs of chromosomes or on that last pair, those sex chromosomes. The words heterozygous, homozygous, and hemizygous that are seen on some genetic test reports tell us about the specific individual who was tested. If the genetic variant was found on 1 copy of a gene, we use the term heterozygous. Homozygous tells us that a person has the change on both copies of their gene, and hemizygous is used to describe a male that has the genetic variant on his only X chromosome. On your report, you might also see the words inherited or de novo, and these terms help us to understand if a genetic variant was inherited from a parent. We determine if a genetic change was inherited or new to a child by testing the parents for that variant. If the variant was passed on, so we see it in mom or in dad, we say that that genetic change was inherited. As many of you might know, some genetic changes are new and unique to a child, meaning that they weren't inherited from mom or dad, but they happened early on in the sperm, in the egg, or early on in development. Whenever a change is new and unique to a child, we call these de novo variants because de novo means new in Latin. So it's an old Latin term that's sort of stuck in genetics, but it just means new and unique to the child. Finally, a few families might have the words mosaicism or mosaic noted on their report. Mosaicism means that only some of a person's cells have the genetic variant. Some cells have the variant, some do not. For some families, this might have been identified through their testing, that when looking at a blood sample or a saliva sample, the genetic testing laboratory only saw the genetic change in some of the cells. With our current testing technology, if we detect mosaicism, we often can't say which cells in the body have the genetic change or really how many cells in the body have the genetic variant. But this is something that might be reported on some people's reports, so I wanted to be sure to mention it. Sometimes we also might come to learn that a parent has a specific type of mosaicism called germline mosaicism. This means that more than one of a mother's egg cells or more than one of a father's sperm cells, what we call their germline, have the genetic variant. Our current technology doesn't allow us to detect germline mosaicism through our testing of blood or saliva samples, But sometimes this is identified when parents have more than one child with the same genetic condition, but they themselves don't have the variant found in their blood or saliva samples. So we might think that the genetic change is new or unique to the child, but we come to learn that a parent has the genetic change in multiple of their sperm or eggs after they have multiple children with the same genetic variant. If a variant is found to be de novo in a child, so we find it in a child, test mom and dad, and neither of them have it in their blood or saliva sample, we say that there's less than a 1% chance for them to have another child with that same genetic change. Again, a family might have more than one child with that genetic change because of this germline mosaicism. So now that we've walked through many genetic concepts in a relatively short amount of time, I want to turn it over to Rebecca to walk through an example report to help put some of what we talked about into a bit more context, to walk through a report with you, to try and drive some of these terms home and help you walk through your laboratory reports. And then of course, we'll be available for questions after we walk through an example.
Great, thank you for that wonderful background, Julianne. So like she said, one of the most common questions that we get here at Searchlight is, how do we read our genetic testing report? And at first look, it can certainly be overwhelming to try to make sense of all these parts and pieces, but we do hope to break it down for you in a meaningful way where all of these parts can be understood. Lab reports from different labs may look slightly different from each other, but they're all made up of the same parts. We'll use an example today from one of the labs we see reports from most often, called GeneDx. And so what's a better place to start than at the top? At the top, you'll see all of the demographic information. So your child's name, birthday, the type of sample that was used. So that may be blood or a buccal sample, which would be like a cheek swab. You'll also see the dates of the whole genetic testing process. So when that sample was collected, when it arrived at the lab, and then when the report was made. And this is important because we're learning more and more about genetics every single day. So if this report is over 2 years old, well then maybe it's time to ask your genetics provider to review it, because we may know more information now than when the report was initially made. And then moving down, the next thing that you'll see is the test that was ordered. And we already covered this, but I'd like to review it just again. There are 3 main types of testing that we see here at Simons Searchlight. The first result that we can see are on chromosomal microarrays. So this is that test that's looking for any missing or extra pieces that we call deletions or duplications. We know that this change in the amount of somebody's DNA can change how their body works and can result in things like medical and developmental changes. You can see here the list of copy number variants that this test would find that we accept here at Simons Searchlight. Moving on, other types of testing we can see are both gene panels and whole exome sequencing. And as a reminder, these are the tests that read through the letters of the DNA. Gene panels are looking at specific genes that we know are related to a specific health or developmental condition. So some people may have had panels looking at genes related to epilepsy or autism and developmental differences. Other individuals may have had whole exome sequencing, and that's reading through all of the letters of all of their genes throughout their DNA. And this type of testing often would result in those sequence changes that we can see in any of the genes listed here. And so now, moving farther down, the next section is often called clinical indication. It may also be called relevant history or pertinent information. And this really just describes why the child is having genetic testing. It's often just a brief summary of their medical and developmental history. And then we move down and we get to the results of the testing. And this is often the media section. So we'll break this down piece by piece to review what all of these different parts mean. So we'll start on the left, and this is where we'll find the gene name. As we know, many genes in our body help our body work like it's supposed to. And this is an important first step in thinking about what any of these changes may do. We think, well, what does this gene do in our body? What instructions does it code for? Many of the genes that we see in Simons Searchlight, for example, may help with brain development and brain growth. And so if we have a spelling change that breaks how this gene is working or changes how it works in our body, it makes sense then that it could lead to things like developmental differences. And then we'll move to the next box, which is labeled disorder or disease. And this tells us the name of the condition that results when somebody has a spelling change in that gene. This name is most helpful if you'd like to learn more about what it means. This is the name that you would type into a search engine, for example, to look up more information. Down in the sections below this box is often a summary and interpretation section. That's when you'll find the most details about what this specific condition actually is and what are those associated signs and symptoms. All right, and continuing on, the next box is labeled mode of inheritance. And for this section, we're jumping back to information we already reviewed today, but I'd like to cover again. So as a reminder, we all have 2 copies of each of our genes. We inherit one from our mothers and one from our fathers. It's totally random what copy we get from each of our parents and ultimately what copy we may pass on to any children. And our body uses both of these copies to work like it's supposed to. There are 3 main types of conditions that we see at Simons Searchlight. The first is called autosomal dominant, and this is most of the changes that we see here. This means that of somebody's 2 copies, 1 has a spelling change, and that's enough to affect how their body is working. We can also see autosomal recessive inheritance, and so this is when both copies of that spelling change are present on both of those genes. Oftentimes, with these autosomal recessive conditions, each parent only has 1 gene change. They're called carriers, and most times, those mothers and fathers don't show us any signs of the condition. It's only when both changes are passed down to the child that we see signs of the genetic changes. And then finally, we have changes that can be inherited on our sex chromosomes, so those X chromosomes. We call that X-linked inheritance, and as we reviewed, males typically show more significant signs and symptoms if they have a gene change on that X chromosome because they don't have that backup X chromosome that females do. All right, so we're going back to our report and making our way through. We'll move on to our next box, which is labeled variant. And this tells us that very specific gene change that we found, that spelling difference that we can think about both in terms of the DNA letter that's changed, but also the difference on the protein level and how that amino acid is changing. And then we'll continue on to zygosity. And this is going back again to that autosomal dominant and recessive. This is telling us how many changes somebody has. So again, if somebody is heterozygous, that means that there's a change on one of their chromosomes. Most often, this relates to autosomal dominant conditions. If somebody is called homozygous, that means they have those 2 gene changes, and that most often corresponds to autosomal recessive conditions. And then when we get to the box that says inherited from, that's really getting at the question of, well, where did this gene change come from? Sometimes these gene changes are inherited from a parent. And depending on what the specific change or variant is, that parent may show us the same signs as their child. They may show us different signs or symptoms that their child doesn't, or they may show us no signs at all. This is also how your provider would be able to tell you, well, what are the chances that any other or future children would have this same genetic change? Many families may be interested in this information for family planning. It's also possible that these gene changes may be brand new, only in the child, and was not inherited from a parent. This is what we call de novo. And in these cases, the chance for any other or future children to have the same change is often quite low. And it's very important to remember with these genetics that these changes are totally random by chance changes that occur. There's nothing that anybody does or doesn't do that causes them. And we also have no control over what DNA we pass on to our children. And then lastly, we get to classification. And this is how your medical providers determine what the change is or isn't doing in somebody's body. And like Julianne reviewed for us, there are 5 main classifications. And these labels just really have to do with how confident are we that this change is the answer for that child's developmental or medical differences. Most often, changes that are benign or likely benign aren't reported because we think that's not our genetic answer. We can also have changes that are called VUSs, or variants of uncertain significance. These are essentially inconclusive results. And these are those genetic changes that we just don't have enough research yet to know exactly what they mean. But we hope that in the future, as we learn more, we'll be able to provide these families additional information as to whether or not this change is that child's answer. And then finally, we have pathogenic and likely pathogenic gene changes. And these are those results that we're confident are changing the way that that child's body is working and is our genetic explanation for why those child has those medical or developmental differences. And then as you look to the bottom of your report, you'll find these usually large sections titled Interpretation and Summary. And this is really where you get a lot of the information about, well, what is this gene doing in the body? And when there are gene changes, what have we seen? What are associated medical findings or developmental differences that we see in other individuals? Your provider will also be able to use this information to guide management moving forward, whether that means seeing other providers or keeping your child enrolled in all of the services and therapies that they're already involved in. All right. And now it is possible that some of our families here today may have had a chromosomal microarray report. And so I wanted to just briefly mention it right here to show you that this is what it looks like. While many of the pieces are similar, it does have some different sections. But I just wanted to bring it up to say that if you have one of these reports, we are more than happy to walk it through with you because it's possible that somebody could have a deletion or a missing section that encompasses one of the genes we're talking about today. So don't hesitate to get in touch with us if you'd like to walk through these parts and pieces as well. All right, and we've made it to the end. So we hope that today's presentation has been helpful in making a little bit more sense and meaning out of these really complex genetics. If you have any additional questions specific to your own child's report, please don't hesitate to reach out to us at our email here, and one of our genetic counselors will be in touch shortly. Thank you for watching.